on a portion of nanobiotechnology, an application part of nanobiotechnology, nanomedicine. As the means majorly this FTP program is for human health welfare. So I thought we'll make use of the new innovations going on in medicine with the intervention of nanotechnology. So a new revolution in human welfare, that is the topic I'll be giving the lecture today. So what is nanoscience? It's the science of tiny things. We have been studying, when I was a student and when was introduced to microbiology, we were fascinated to see microbes under the scanning electron microscope or TEM, and to see their such tiny organisms giving us such wonderful science. And so when we see microbes and that time of the 3 or 4 decades ago and coming to nano world now, we are into another fascinating world. So let us see a little bit of the fascinating world of nanotech. And take example, a human fingernail grows 1 nanometer every second. And can you imagine a man's beard grows 5 nanometers every second? As the participant is sitting there, his beard is growing 5 nanometer each second. So, and this picture shows a silver nanowire, silver nanowire resting on a human hair. Human hair itself is so delicate and thin. Just imagine if you magnify, this silver nanowire will be so small. It's just 2 nanometer in length. So when I want to introduce about nanomedicine, it's not new to India. Our ethnomedicine has given us lot of similar medicines ages ago. We have all known that basmas, an Ayurvedic metallic mineral preparation, is claimed to be prepared of biologically produced nanoparticles, making use of the biologically available natural products and preparing medication. And that was ethnomedicine, wherein our Ayurveda, homeopathy, and Unani is known. So, but as a science of nanotech came, the room of the opening of nanotech started with the father of nanotech, Richard Feynman. In 1959, December 29th, he had attended an international seminar wherein it gave idea of what is
ಕೇಳ್ಕೊಂಬಂದಿರೋದ ಕೇಳಲ್ಲ ಎಷ್ಟು ಚೆನ್ನಾಗಿ ಅವ್ರು ಹೇಳ್ಕೊಟ್ಟಿದ್ದಾರೆ.
Hello, please mute. And so there's plenty of room at the bottom. That's the sentence is stated. There's plenty of room at the bottom. And this lecture and the ideas he narrated about nanotechnology gave the idea for the field of nanotechnology. Hence, he was given a Nobel Prize in Physics in 1965, and he is called father of nanotech. And it was a Japanese scientist, Nario Taniguchi, in 1974, who coined the term nanotechnology, and it took almost a decade for Harry Kroto to prepare a real nanomaterial. He was the one who invented a buckyball, a fullerene carbon atom ball, hexagonal shape, spears mixed to make a ball, for this discovery, he was given Nobel Prize in Chemistry in 1996. And later on, it was Erich Drexler in 1986 who popularized molecular nanotechnology. So when did nanotechnology really start? When you see ancient history, and if you visit European, like your Roman Catholic churches, or if you've seen the paintings, you would have seen all of their ceilings and walls with beautiful metallic paintings, which are having glowing inks. So now we realize these inks are nothing but silver and gold nanoparticles of various sizes. So also, if you know that famous Licaris cup, which is a 4th century Roman glass cup, it glows differently. In dark, it is not visible at all. But when you put red light, when red light is coming from behind of the cup, it glows red. From the front, it glows green, as it's seen here, because of the specific nanoparticles which are filled inside the glass. So how is this working is, Nano is a very small material. What we are trying to do is take a bulk material and cut its down, its size into very minute shape, so that we increase the surface area to volume ratio, which encourages us to do this kind of wonderful, what we call as art now is turning into science. How is it useful? Take example, when we have a sugar cube and granulated sugar, which does dissolve faster? Of course, granulated sugar. That's the role. From a bulk material to a granulated sugar, there is a difference in dissolving. That's what is the purpose when you want to design a drug. Instead of giving in bulk, we are giving it as in a minute quantity so that its dissolves and bioavailability as well as targeted delivery occurs naturally. So take example of how these matters are working differently at different size. With the best example of gold. We all know the color of gold is shiny yellow. But did we know that it at various nanoparticles, gold is appearing in different color? It ranges from red to maroon to blue to purple with changes in the size. Lower the size, it is red. Higher the size of the material, it is purple. So different size of particle reflect and absorb differently. So that's what is the result of the quantum effects, which dominate the behavior of matter at nanoscale. For example, your bulk gold, which is shiny metal, when you cut down its size into different nanometers, 4 nanometer, 15 nanometer, 60, 100. You can see 4, it is red. When it is pure bulk gold, it is yellow. 4 nanometer, it's red. 15, it is dark red. 60, dark pink. 100, it is blue. So like that, when you see, you can see the differences here. The small nanoparticle appear in red, while slightly bigger appear in blue. This is how your different particle size of gold are appearing because of the color change because of the size. That's what you see in this metallic paintings. Here you see in this glass painting in the Roman Catholics, you see red stained glass. That is when the gold is 20 nanometer in stage. Whereas you see orange tinges in his gown and also in this plate, it is 80 nanometer gold. So, and yellow here, that is pure dominant gold. So like that, the dilutions and the changes in nanomaterial gives us different coloration. That's what we are looking for when we are going in for nano imaging or nano diagnostics of various diseases. So let us see some of the natural gallery of nanomaterials. This is a SEM image of a butterfly wing scale, and you can see how the nanostructures look. These give us ideas to come up with biomimetics. Another best natural example is lotus effect. We all know this, our national flower, lotus. Though it kesaralli builded, we say it grows in dirt, but its leaf is always clean and clean, pure clean, though it is growing in dirt. Why? Because of its unique capability of self-cleaning. That is because the upper surface of the leaf have ultra high hydrophobicity because of the architecture of the nanostructures of the leaf, which rolls in the dirt water and removes away the droplets, that keeping the surface area completely clean. So that's one of the best example. And also you can see another natural example, how a gecko, when it jumps on a glass and starts climbing a slippery glass wall also. See, because it has its nanostructures, which gives a grip for it to put its hand and feet also on the plane of a glass which is slippery. So that is because of the nanostructures which encourage them. So all these ideas we get from nature. Even a peacock feather, the minute color changes in between its own single hair, how it is different. It's because of the nanostructured color vibrations. So also when we means before we all were even born, Hollywood came up with a conceptual movie a future of science, nanoscience with a film titled Fantastic Voyage. It was all an idea, visionized idea in 1996. And here you can see how a robot is running through the blood cells, through our endocrine systems. So that is coming into reality as these robots in the bloodstream looks for which is the blood cell, which is having cancer, which is healthy cell, and tries to block or kill or damage the cancerous cell only. That is what nanotechnology with the blend of biological sciences like biotechnology is giving us idea with the intervention of nanobiotechnology. So our great president, late president, Dr. APJ Abdul Kalam, over a decade had stated, to understand the very large, we must understand the very small. Nanobiotechnology will be the future with nanoscience developing nanomaterials as well as devices. This will lead to further converge of technology with wide application. So that was his future thoughts and focused idea. And so now we have nanobiotechnology, which we call as definitely the next revolution in the world of science and development. So what is nanobiotechnology? Biotechnology as a science is the application of technological innovations specifically for life sciences. Whereas nanobiotechnology, it incorporates the life science techniques with at the making applying at the nanoscale level. So nanotechnology blends with biological science to give us nanobiotechnology. So how are we preparing this tiny useful nanoparticles? We have two approaches, top-down approach as well as bottom-up. Top-down is we are taking a bulk material or fragment, and then we are preparing them in cutting down to nano sizes from 2 millimeter and then to nanoscale structure. Whereas bottom-up is we are taking the 1 nanometer material and clustering them and adding other kind of substances and making them into useful organism. It might be atoms, clusters, and nanoscale devices. So when we're starting from the nanoscale, it is bottom up. When we're starting from a bulk above like a millimeter or meter scale, that is top up. So how can we do that? We have physical methods, chemical methods, and biological method. The first to be invented by physicists were the various physical techniques of preparing nanomaterials like ultrasonication, irradiation, microwave, and electrochemical science. Whereas chemically we can do by sol gel method, chemical reduction, inert condensation methods. Whereas though these two methods are good, they come up with certain type of usage of certain ultraviolet rays or very toxic chemicals. Hence, we look for the very important non-toxic methods like biological methods, wherein we're making use of the naturally occurring bacteria, fungi, or algae, or yeast, or actinomycetes, that is the microbes which are being used as a starting material for synthesis, or the plant-based, that is phyto-based synthesis or phytofabrication of nanomaterials. So where did we all get this idea? Definitely from nature, and that is biology, the inspiration for nanomaterial synthesis. You can take a rice blade example or radiodolurans or your diatoms. Their natural look itself is like a nanostructure when you zoom and see. They give us ideas how we can prepare various nanobiomimetics. Now, all these ideas, the scientists, nanoscientists are preparing various kind of scaffolds, even for your bone stem cell regeneration or arthritic issues, or even for your heart transplant, patch up cells for mesenchymal stem cells to regenerate in your heart region. So all this is the science which is developed over the last 2 decades with all ideas from nature. So when you're trying to do biogenic synthesis of nanoparticles, we know that nature acts itself as biolaboratory, and the plants, the algae, fungi, yeast, which are each one of these organisms, the plants, the algae, fungi, they have their own kind of important biomolecules. Like example, our yeast, they are having curdlans or beta-1,3 glucans, which act as like a capping agent. Fungi have their own kind of chitin and chitosan and oligosaccharides. Algae is rich with such amount of PUFAs, and plants are having their own kind of different enzymes, and they are very rich in secondary metabolites, which act as a capping agent. So making use of this innovative their self-synthesizing capability, we are trying to make use of these biological systems to synthesize our nanomaterial, which is of useful for either for medicine, environment, or any other area. So what we are trying to do for biogenic synthesis is we're involving bioreduction of metal salts into elemental metal. So we are taking any of the biological system, be it your plants or algae, diatoms, bacteria, yeast, fungi, or even human cells as starting material, any one of them. And when we are taking, like if you're taking fungi, we are taking the fungal extract or mycelial extract or culture filtrate. To that, like you take example fungi, the material might be trichoderma. Trichoderma can be taken as mycelia or even the culture filtrate. So to that, we add the precursor for that particular metal, be it your zinc or selenium, silver or gold. So each one of them have their own precursor metal. So when you add the precursor along with the live cells, they transform inorganic metals into metal nanoparticles. So, and because they have their own bioreductive capacities, that is their own proteins or enzymes or other biomolecules which bring about the bioreduction. So like that, these are the other different kinds of biological systems which are able to synthesize gold or your iron or silver, whatever the metallic nanoparticles we need. Bacteria can synthesize both extracellularly and intercellularly. Fungi and yeast can synthesize both extra and intracellular, as well as they can synthesize through cell wall surface. Plant extracellular and also very inexpensive reduction in the simple mode. Many of them prefer even for plant-based, and next comes fungal-based. So when you see what are the different types of nanoparticles or nanomaterials we can synthesize into, it can be organic nanocarriers, inorganic nanocarriers, and biological nanocarriers. Biological, it can be viral vector carriers or extracellular vesicles. It is inorganic, it can be inorganic nanoparticles like we discussed, the metallic ones, carbon nanotubes or quantum dots. And when you see organic, it is polymeric, solid lipid nanoparticles, liposomes, dendrimers, micelles. All these are having very much useful role in nanomedicine. So when you see them in the devices prepared through this synthesis, the nanoscale they'll be measuring, will be all these devices not more than 10 nanometer. So what is the scale of 1 nanometer? It measures 1 into 10 to the power of minus 9 meters. So nano means dwarf in Greek, and you can see the scale here, 10 to the power of 1 minus 1 to 10 to the power of 8. 10 to the power of 8 is your tennis ball. 10 to the power of minus 1 is water molecule, and our nano devices, whatever we try to synthesize for nanomedicine, it is either your nanopores, dendrimers, nanotubes, quantum dots. All of them will be lesser than 10 to the power of 2. So they are so tiny. Okay, so what is the advantage of preparing this kind of nanoparticles? Their process, once you standardize, is very simple, very cost effective, because we are not buying any expensive chemicals or chemical physical synthesis. It's all simple plant or fungi or algae, eco-friendly, easy availability, zero contamination. And as we are using biological organisms, their own proteins, their own metabolites act as capping and reductive agents, and they have high reducing potential, easily scalable, and also we can easily fine tune with the help of all the innovative methods. So what are the innovative applications of this such kind of nanomaterials? Nowadays we are having nano fabrics. Their role is there even in cosmetics, bioengineering, defense and security, optical engineering, and nanobiotechnology, energy, as well as medicine and drugs. Today, we'll be focusing majorly on nanomedicine. So nanomedicine refers to highly specific medical intervention at the molecular level for curing disease or repairing damaged tissues. So though this science is very much in its infancy, just since 15-20 years, the research is going on. Very few have been gone through all the phases of clinical trials and released into market. But last 4-5 years, lot of research has been going on to make the maximum benefit from this nanomedicine science. So when you see the role of it, it's not involving only one kind of role, not only a medicine person, but it is having intervention from a biological scientist, nanotechnologist, and chemist, a pharmacologist who prepares the drug. So it's a common interdisciplinary field which has biology, nanotech, and chemistry working together. So when you see the definition of nanomedicine, it's defined as monitoring, repair, construction, and control of human biological systems at the molecular level using engineered nanodevices and nanostructures. So also you can say in another way that the detection and controlled manipulation of human biological system at the molecular level, making use of engineered and nanodevices, that is ENDs or engineered nanostructures. Like it might be a nanorobot or a dendrimer or a nanotube or your nanoshell. All these are the various nano devices. So what are the various nanotechnology application in medicine? They are divided into 3 parts: nanodiagnostics, nanotherapeutics, and immunization and vaccine production. Which immunization and vaccine production, the research is still started now. Whereas the nanodiagnostics and nanotherapeutics, there is already research, and the work is already taken to the bedside also. And like we are having a lot of diagnostic apparatus, our MRIs, our cancer detecting tools, all equipped with nano devices as imaging agents for cancer diagnosis, for diagnosing infectious as well as non-infectious diseases, and for treating infectious and when you take therapeutics, it will be useful for treating infectious and non-infectious diseases, for cancer treatment, gene and stem cell therapy and regenerative medicine, tissue engineering, and prosthetic orthopedic implants. Where a lot of research and orthopedic, especially preparing nanoscaffolds for orthopedic implants is going on, and we are getting very light material nanoscaffolds which dissolve inside, and they're very helpful in orthopedic healing. So let us take up nanodiagnostics. So how the nanotechnology is encouraging diagnostics in medical field. So earlier, if you see a decade earlier, if we had our MRI scans or X-ray scans, the visibility was like in 1 meters. Now, like this whole globe, 1 meter, when you compare it with 1 nanometer range. So the tiniest infected cell or tiniest cancerous cell can be diagnosed easily. So that's where nanotechnology is having a big role in medical diagnosis and therapeutics. So it provides new materials with novel optical properties and for various biomedical applications, majorly in diagnostics, drug delivery, tissue engineering, and biosensors. And what are the various nano devices used in diagnostics? Cantilevers, nanopores, nanotubes, quantum dots, nanoshells, dendrimers, and magnetic nanoparticles. So what is the beauty of these devices? Each name coming with a different kind of terms. All of them have the potential basic property of having a unique melting point and having an optical coloration. And more importantly, the pathologists are more interested to find out the changes in potential increase in the physical size of each molecular component. And based on the molecular components, decreased or increased size can be visualized. And another important thing is that an increase in fraction of molecule of surface of the particle will change with surface reactivity. So this encourages us to come up with early and accurate diagnosis of the disease. Why? Because we are tagging that particular nano device with the biomarker, which goes and attaches to only the infected or diseased cell, not the healthy cell. And based on the biomarker fluorescence or illumination, we can say what stage of that particular infection or the cancer is. So this is one kind of non-invasive and painless diagnostic technique. And this is also encouraged genetic testing for individual with personalized therapy and personalized medication can be given. So where it is taking a very great jump is in cancer diagnostics. So when you go to a medical doctor or oncologist, he will try to examine where all you have the pain and think, oh, that you might, based on the symptoms, he might think your pain is here, like this green dots, and the area around your, like your kidney. So what later on, when you go for imaging, it will come down to one area where the pain is. But with the nano imaging or nano diagnostic, we'll be accurately pointing out which is that particular organ, tissue, or site you have the infection or the cancer growth. That's how it's accurate and smart. Why? Because we have such tiny nano devices. They are so small enough that they can enter easily a tiny cell. So this is the nano device. Take example, it might be a dendrimer, which is measuring only 10 to the power of 2. But as a nano diagnostic tool, it can easily go there, as it is bound with a biomarker, identify the disease cell. So one such very important discovery is this beautiful QDIS, which we call them as quantum dots, a very minuscule molecule which is used to detect cancers. So it's, what are they? Quantum dots are the tiny crystals. They glow differently. Each crystal glows, as they vary in size, glow differently in ultraviolet. So that's what we are trying to make use of for detection of cancer. And the latex beads filled with these crystals can be designed to bind specific DNA sequences. So when these crystals are stimulated by UV light, each bead light that serves as sort of spectra barcode identifies the region of the DNA. So that's how it's been used. So quantum dots, if you want to name it in a single word, it's a fluorescent nanocrystal. And common type of quantum dots, it can be your cadmium, selenium, or lead selenium, or sulfur or copper chloride based. And they are also called as artificial atoms. Because of the wonderful, though they are chemically synthesized, they have wonderful role to play in vitro imaging, in vitro targeting, bioanalytical assay, as well as biosensors. And when we make use of them as nanobiosensors. What is the unique property of quantum dots is they are biocompatible, they have excellent water solubility, low toxicity, and the major focus on these carbon-based quantums is because of the way they can have good solubility and strong luminance. We call them as carbon nanolights. So that is another way of calling them. And how they are designed is the major property of these quantum dots is they are prepared as hydrophobic crystals, and at different size from 0 to 10, maximum being 10 nanometer. But how do you make them biologically stable? We are making a hydrophilic coating with a biopolymer or biological substance, and tagging them with the protein or the biomarker so that they can identify the site of infection. So that's how the diagnostic is made easy. We call it as diagnosis by a dot. So quantum dots stick to the tumor in a mouse thigh, like you see here, glows red under infrared light. So this is simple non-invasive method of how you can diagnose. So how is it working? Just like, take example, you have your quantum dots here. These are the quantum dot beads. light is off, there is no fluorescence. Once UV light is on, they emit light. Each this crystal of different size, that is 1 to 10 nanometer, emit different lights, different color in light, which is absorbed, and you can find out based on the intensity what is the stage of that particular tumor or cancer. So that's how we are trying to, this you can see quantum dot. It is having a CDSC core, zinc sulfate shell, and topo coating. So, and this is a 4 nanometer dot which is prepared with different colorations, you can see, and they are used as to find cancer signatures. So these are the cancer cells in amidst the healthy cell. What happens is when you go in for UV elimination, only the cancer cells glow, whereas the healthy cells are not glowing. That is how they detect, and imaging is made easier. So you can see how the tumor in different, this is the various stages of the tumor detected in the mouse. I've used as a model, and image in a live mouse, how you can use quantum dots for imaging. So that is where the major way quantum dots is going in imaging. Next is the MNPs, the magnetic nanoparticles. They are having multiple role in multiple disciplines like MRIs, magnetic resonance imaging, nanoelectronics, and biosensors. The basic elements of MNPs are that they have iron, nickel, and their derivatives in nanoparticle range, and hence they are acting as versatile diagnostic tool, and they bring about in vivo MRI imaging to or bring them into a greater effect. So in the upper application of these magnetic nanoparticles with the invention of spions. Spions are super paramagnetic iron oxide nanoparticles made up of iron oxide core and coated by any of the inorganic materials, like it might be silica, or organic materials like natural polymers, dextrans or chitosan. So as these spions act as versatile agent and are involved majorly, now they are very good markers for detection of atherosclerosis. So what is that is the spions are acting as contrast agents for MRI imaging and encourage the sensitivity and selectivity in diagnosing issues like ailments like atherosclerosis and in various types of cancers, and even in their regenerative medicine it's been used. So another important material which is taking all the limelight this decade is graphene oxide. It is GO, that is graphene oxide, is a very thin layer of hybridized carbon, very much having a medical diagnostic role and exciting properties. A very thin sheet of graphene oxide is attached. It is a thin sheet made of hexagons of carbon atoms, is attached to antibodies, and these sheets go and carry the cancer cells on them, and as they're tagged with the fluorescent antibodies, they glow and encourage the detection of cancer cells, 3 to 5 cancer cells in 1 millimeter of blood sample. So especially useful in detecting blood cancers in a very early stage. So gold and nanoparticles are the metallic nanoparticles which are having multiple uses in diagnosis. Gold nanoparticles extensively used in diagnosis of cancers, and silver in majorly infectious diseases and labeling and biosensing. They're very much used. And what are the different contrasting agents used in MRIs? We see granulofullerenes, the carbon atoms, the dendrimers, missiles, carbon nanotubes, all of them are being very well used in the diagnostics. So you can see here how these various types of fluorescing diagnostic tools encourage to distinguish between a healthy cell and a cancerous cell. The healthy cell is not glowing, whereas the breast cancer cell fluorescence, and with minimal fluorescence, you can detect the cancerous cells. So that's how nanodiagnostics is taking up a huge role, especially a lot of equipments nowadays are having nano devices, especially the MRIs and the tools which are used for cancer diagnostics. They're having the nanostructural devices. So now the next is the therapeutics. What are the applications of nanotechnology in medicine? We are going now into nanotherapeutics. So why nanotherapeutics? Because we want, like we take one kind of antibiotic, which like we have food poisoning and the target is our intestine, but the effect will be throughout the body. That is the kind of the regular traditional medicine we have. But what we are looking at here is controlled release of drugs, targeted release of drugs, smaller and faster devices, and without much side effects. So that kind of drugs we are trying to look. And what were the various type of drug delivery systems we had from the beginning? We are known to take medicine as oral or injection based or transdermal, like your bandages, and carrier based. So now all these give kind of a complete body treatment. What we are trying to do is now develop a treatment for that particular organ or tissue or inflamed cell. So that is the target, and not at a single go. We need the capsule or the drug or device to deliver medicine smartly, control the release capacity based on the need, based on the symptom. I have fever now, but I take the medicine. That immediately the fever is getting reduced. But after 2 hours, again I get a fever. That is because we understand the infection is there again. This controlled release will be so that manipulating the thing that there is temperature increase, the medicine is again released. So this device senses the change in the temperature and releases again. So that's how individualized the therapy can be done and monitor even drug absorption and distribution and metabolism. So like that, now instead of tablets and capsules, last decade we also have seen thin film drug delivery, wherein instead of taking the drug orally, we take a small drug strip and keep it on the tongue and so like you see a neoarthrum drug strip here. This strip is given for various infections. You just have to take the strip and keep it on the tongue. It melts and the drug slowly dissolves and absorbs through sublingual. So like that, we also have hydrogels. Like when they are prepared, they'll be tiny like this. But when they are imbibed and they reach the intestine, they imbibe and glow or come to an original size. So that's how the hydrogels work. And that is from the classical drug. Now we're moving through nanodrugs. So when you see that classical drug, the dispersion, like the problem here is inflamed colon. The colon is the only one which is having inflammation. So, but the medicine what we take in classical drug is distributed throughout the body, and the concentration of drug reaching the colon is reduced. Whereas nanodrug, the target, it's reaching directly as it is going to bind to the inflamed cell receptor. It is binding there and targeting only the inflamed drug, colon. So that's what is the new science of nanomedicine. The new science is targeted drug delivery. So let us see what is target drug delivery is. The nanoparticles containing the drugs are coated with specific kind of the targeting agent. It is majorly the conjugated antibodies, and this, as they have conjugated with the antibody, this search for the kind of the signal which is coming from a cancerous cell. It goes and binds to that kind of cancer cell or an inflamed cell receptor, and then releases the drug to that particular site. So engineered nanomaterials, that is ENMs, they range from 1 to 100 nanometer, and they are having properties at the size of nanomaterial. Nano size, that is up within 100 nanometer. Most of these drugs are prepared within 10 nanometers. These nanocarriers for targeted drug delivery can be metal-based, lipid-based, polymer-based, or biological nanoparticles. So when you see them, these nanotools, which we also call as nanocarriers, they can be nanorobots, nanoshells, quantum dots, dendrimers, carbon nanotube, nanowire, nanosensors, or nanoliposomes, or you also call them neosomes. So you can see how these devices look: quantum dots, liposomes, iron oxide nanoparticles, carbon nanotubes, gold nanoparticles, polymeric nanoparticles, dendrimers, micro and nanobulbs, upconverting nanoparticles, iron platinum nanoparticles, nanoclusters, and functionalized nanoparticles. So all of them having various role in detecting various infectious and cancerous diseases. So each of these nanocarrier have the role of carrying the drug in the small size and delivering the drug to that particular site. And what is the major role of the biological property is their entire surface area allocates the availability of or absorption availability of the drug. So the permeation of the drug across epithelial endothelial barrier is easier because they are so tiny, and to deliver the drugs at the site of action. And they're having combined therapy, that is they're encouraging diagnostics of that particular cell as well as curing that particular cell or killing that cell if it is diseased. So again, you can see the different kinds of types of nanoparticle: polymeric missiles, liposomes, biodegradable or biocompatible nanoparticles, nanoparticle drug carriers, dendrimers, nanospheres, artificial DNA nanostructure, or stimuli-based drug-releasing nanoparticle, silica nanoparticles, metal nanoparticles, carbon nanoparticles, magnetic nanoparticles, nanocomposite, which has one kind of organic material along with metallic nanoparticle. It's a blend of one-dimensional and two-dimensional nanomaterials, and protein, which is conjugated to protein drug nanoconjugates. So let us see all these nano devices and their role in targeted drug delivery. The very first one to be seeing having very great benefit is C60, or you can say the carbon 60 carbon atom, which is just 1 nanometer diameter. It's arranged into 20 hexagons and 12 pentagons. It's like a tiny 1 nanometer football. It's just the shape of a football. And nanopharmaceuticals were the first to make use of it as a drug delivering agent. So you can look at this 3D image of the C60, one kind of a fullerene. As you see, this is nothing but a nano cage. Inside this cage, you can trap your drug, and outside you can tag it with various antibodies. And this acts as a marker and attaches to that particular disease cell, and then the drug is released. So that's the beautiful role of fullerene. And you can see one modified fullerene called as modified buckyball, and they deliver radioactive atoms to cancerous material, that is C60, against CA colon cancer. So transfer of radiation is within the ball, hence minimize strong radiation to the healthy tissue. They go inside the infected cell, and they radiate and then bombard the cell. So it is not touching or going anywhere near the healthy cell. So that's how this buckyball based drug delivery system works. And you have nano robots, these nanoscience machines, which we saw in that movie as a device. And very much important to detect our blood means blood cancer cells in a floating endocrine system. Nanoscience machine, they run through the endocrine system, and then they amidst the various RBCs, they try to find which is the cancerous cells. So these are the devices which do that. Nano robots, and also nanotubes, nanowires, all of them one-dimensional or two-dimensional nanostructured materials used to carry drugs and gene delivery systems. So they are all like your single fullerene is in round. The same 60 carbon atom can be made into tubes and sheets like our graphene oxide sheets, or tubes like nanotubes or nanowires. They're prepared like that, and they're made use of it. Like you can see the another nano robot, which is, you can say, teeth cleaning robot, which kills you have teeth or our oral cavities have a lot of issues with repeated infections and causing of biofilms. With repeated antibiotics also, these biofilms are not dissolved. But when you give treatment with this teeth clinic or the gum cleaning robots, they go and break these nanofilms. Similarly, especially we have seen the last 2 months, a lot of coughing issues due to pneumonia and a lot of lung pulmonary inflammations I've seen because of this infections are deep inside our lung cells and wherein the medicine is not able to penetrate. Hence, the prolonged coughing is there because of the severe pulmonary inflammation. But such nanorobots can go into that particular site of the bacteria where it is adhered and called biofilm and kill them up and open up the phlegms. So that's why you have called them as lung cleaning robots are inhaled where nanorobots can collect foreign particles. Most of them even are allergens which causes pulmonary inflammations. So they can remove those allergens like fibers or asbestos and encourage killing of them. Nowadays, we also have even robot cleaning arteries, wherein the cholesterol is the major culprit of causing blockages of arteries. Cholesterols can be easily dissolved with this nano robot, which cleans up the arteries. So all these are the ones which the nano drug delivering systems. They can be vehicles can be act as a lipid vehicle or cargo vehicle and or your liposomes which are easily dissolving and which have various surface sugars or prevent destruction by the which can be by the immune system, or they can carry surface proteins to target liposome to specific location in the body, or solid to water soluble drugs in the core and all soluble drug in the bilayer. So all of liposome acts as a solid vehicle carrier of drugs, nano drugs. So earlier we used to take all kind of injections, intravenous, intramuscular, subcutaneous, interstitial, top, and now all those are painful. From the painful kind of delivery systems, we have come to the total non-invasive and non-painful targeted drug deliveries age of delivering medicine. That is, we call it as, otherwise earlier it was passive targeting. Now it is active targeting, wherein only the disease cell is being treated, not the whole organism. As we are tagging it with specific antibody, which goes and binds to its specific signal on the receptor and causes active targeting and delivery of the medicine, this affinity ligands, whether it is antibody or DNA or RNA, are attached to nanoparticle surface, and these nano devices are carried to that particular site and delivered. Hence, active deliveries, an active way of delivering the targeted delivery is taking the traditional drug delivery to a different level of dosage release, specificity of release, efficacy of release without any side effects. So what is the major difference between nano versus traditional drug delivery? The major thing is the controlled and smart release based on the need, and the rest of it is there within the nano device. The little of the amount of the drug is released based on the symptoms and very much targeted to that particular site and very effective. So, and hence we do not need that particular huge dosage which the traditional medicine was giving. A lower dosage and targeted active delivery system is encouraging simpler curing and treatment of various devices. So one area wherein nanomedicine is taking a very great step is cancer chemotherapy. We all know when 2 to 3 decades ago, when cancer treatment was given, we all know how a patient was suffering when through chemotherapy because the side effects of the various, not because of the thing that the cancerous cells were dead, because the chemical agents which were using as chemotherapeutics, it was killing not only the cancer cells, also the healthy cells. You can see 50% of healthy cell, 50% of cancerous cells killed with the because of the regular chemotherapy. As now the targeted drug delivery is majorly aiming only for the infected, that is the cancerous cell, the healthy tissues remain as such, and hence there is less kind of damage to the healthy cells. Why? Because the nanoparticles containing drugs are coated with targeting agent, that is conjugated antibodies, which circulate through the blood vessels and reach the target cells. And major property of these nano devices is because of the controlled release profiles, especially for sensitive drugs, long-lived, biocompatible, and inexpensive, and they are mechanically strong, inert, and they are not, they are also safe from accidental release, and they are having the controlled and smart release effect. So let us see few of the examples of these drug delivery nano devices. One of the simplest, very beautiful device, the tiny device discovered is the dendrimers. So this is how the dendrimer look. Dendron, it's getting the name from the dendron, which means a branch of a tree. You can see it's like of a tree. You take a picture, it looks like a dendrimer. From a single root, the branching out is seen in 3 branches. So they're also called as arborols or cascade molecules, and they are highly branched 3-dimensional molecules, and they are highly defined dendrimers. So as a single, it will be seen like this. When you cut into one core, this is the hard shell, and these are the 3 layers of 3 branches. And you have so different areas you can target the drug. Like you can have the drug antibody conjugates at one time, drug one side, and the various other biological markers which lead to the target cell easily. So nano devices has a link between detection, diagnosis, and treatment. So traditional cancer treatment is you have a cancer cell, you just take an oral medicine or intravenous medicine as drug, and it acts on all cells. Whereas nanobiotechnology based cancer treatment, the nano device, take this as a dendrimer, is entering the cell. Not only, see what all it is having here: imaging, detection, targeting, reporting. So imaging and detection occurs, reporting does, then the targeting is active. What is the targeting? The drug delivery. So each of this nano device can improve the sensitivity to detect. In one small square, you can see there are normal cells, precancerous cells, and cancerous cells. So when you add your nano device can enter cell and distinguish between all these 3, normal, precancerous, as well as cancerous cells. So improving cancer treatment, you can see the traditional treatment, the drug carries, kills the cancer cells, and these are the non-cancerous. What happens? Both of them, not all, at least few of the non-cancerous are also killed along with the cancerous cells. Whereas the nano devices, as their as the cancer cells release toxin, they act as an antigen, and the antibody coated nano device bind to them and attack that particular cell, report that cell, and then deliver the drug to that cell, and hence all the cancer cells are killed, whereas the healthy cells are smiling and intact. So that's how your dendrimers are acting in diagnostic and therapy of cancer cell. They are acting as cancer detector, the detector molecule on one branch, and then the reporter on another branch, and therapeutic agent on another branch, and the total cell death apoptosis mechanism recorded on the other branch. So that's how one single nanodendromer can bring about the whole process of killing a cancerous cell. So now the big lead in cancer treatment is also taken up by gold nanoparticles. They're modified into either rods, multi-rod stars, or in the hollow structures such as shells, nanobox, or nanocages. And they are having very much improved in field of drug delivery, especially in photothermal therapy in oncology, because of their unique photothermal property. So you can see what is the major application is selective nanophotothermolysis. That is laser-induced thermal explosion of gold nanoparticle. What is it is your gold particle, nanoparticle from 1 to 10 nanometer is individually taken, and the surface is functionalized. Surface is functionalized and coated with the drug as well as the biomarker. So, and or else even that they are tagged with a specific fluorescent dye. Selective nanophotothermolysis is the process what we call, and this is giving a lot of research opportunities. Their researchers have shown how gold nanoparticles can be used in cancer diagnostics to bind them to only the malignant cells and not the non-cancerous cells. They're for promising candidates for photothermolysis because they are strong absorbers, they are photostable, non-toxic, easily conjugated to antibodies or proteins, and they have adjustable optical properties. Here you can see an image of gold nanoparticle sticking to cancer cells and which make them shine. One such very important one which is already in use is gold nanoshells. Gold nanoshells, this is the one is shown in the image, have metallic outer cover layer and silica as core. So this is the 50 nanometer metallic gold nano cover, silica core, and it targets either through EPR or small coating on shell, and nanoshells are heated with external energy, killing the cancer cell. How it works? The nanocell shell will let us see now. So this is the near infrared light, which is off. We have your gold nanoshell here. When the infrared light is on, nanoshell absorbs heat and blasts open. And this is doing when it reaches the cancer cell. It's like a suicidal bomb, reaching the cancer cell and blasting it. So that's how it's working. So here you can see the nanoshells. Here you have cancer cells and healthy cells. And as nanoshells are coated with antibody, they come and bind to the specific antigens on the cancer cells. And when you put near infrared light, it immediately blasts the dead cancer cells, whereas the healthy cells remain intact. So that's the way, one little bit of glimpse I'm giving for the cancer treatment therapeutics, making use of nanomedicine. Let us see a few of the other applications by the nanomedicine. One of the important thing is we undergo various orthopedic surgeries or the bone related surgeries. After post-surgery, there are some kind of inflammations or infections. And what we call it as bone infections or bone or osteomyelitis. Osteomyelitis is caused by Staphylococcus aureus, which colonizes intracellularly, internally, anaerobically, and cause a lot of inflammation. You cannot give a treatment to it easily. So you can see in between the bone cells, it goes there and cause inflammations. In interior, very interior. Treating it is very difficult. So there is now the treatment making use of nano devices prepared from or a nanocomposite, chitosan, morley composite, nanospheres for sustained antibiotic delivery. So this is an infection which cannot be taken like we take antibiotic as a course, but that infection will not be dissolved or cured within one course. So hence, based on the need, a sustained antibiotic delivery is done to the post-implantation bone infection treatment. This was published in Biomedical Materials in 2019, worked by Kimna et al. And they've shown that how such kind of important surgery-related infections can also be cured by nanomedicine. Nanomedicine, another huge role it is playing is to encourage treatment of one of the most devastating diseases of nowadays, the neurodegenerative diseases in aged people. We have now nano robots which are designed to move across BBB, blood-brain barrier. Yes, these tiny devices can deliver drug into an inflamed cell of your Parkinson's disease or Alzheimer's disease. So the last decade has seen a lot of research in brain studies, in nano devices, in diagnosing brain disorders as well as neurodegenerative disease. So the same nanocarriers which we studied, it might be your gold nanoparticles, carbon nanotubes, your dendrimers, nanoemulsions, or your polymeric nanoparticles, liposomes, are used to deliver drugs to either early diagnosis of your Parkinson's or Alzheimer's, or even cure them. Like one of the important study shows that zinc oxide nanowires are used to treat Parkinson's disease. The detection takes longer time. By the time we detect these diseases, they'll be in an advanced stage and treatment will be very difficult. The zinc oxide nanowires can detect in changes in uric acid level in a very early stage and encourage detection as well as cure. Because it is coated with three-dimensional hierarchical graphene foam and attached to biomarker, and easy early diagnosis of Parkinson's disease is possible. Similarly, Alzheimer's disease also, we know it's very difficult to treat because whatever drugs we have cannot pass the blood-brain barrier and reach that particular site of plaque formation, beta amyloid plaque formation caused because of Alzheimer's disease. So the nano devices have created a set of environment, a contrast agent, AD drug, that is Alzheimer's drug, nanoparticle coated with alpha beta peptide and important antibody coating. And this is how formulation is done. You can see this beautiful formulation of a nanoliposome, also called as neosome, and which is already in use for Alzheimer's disease treatment. So this single nanoliposome is modified specifically targeting a brain cell. It is pegylated, peg glucose bound. It is mixed with curcumin, lipid peg curcumin compound that is easy way by curcumin is also a novel Alzheimer's treatment. It's given there and it's tagged with DNA, lipophilic drug, protein peptide, hydrophilic drug, and transferrin. And how is it moved inside the blood-brain barrier? Receptor mediated transcytosis. So that's how it moves it, because it is so tiny, it can be easily moved there, or else it can also moved in by carrier mediated transcytosis, and it enters the cell and looks for the beta amyloid plaque. How does it identify? Because it senses the antigen on the plaque, because it has specific antibody, binds on it, liposome binds on beta amyloid plaque and cause disintegration of the plaque. So plaque dissolved to a greater extent. That's how we are targeting even the Alzheimer's disease cure. Okay, and now nanomedicine is taking great role even in tissue engineering, especially in bone tissue engineering, and a lot of scaffolds have been used. You can see how the mesenchymal stem cells are harvested from human or animal souls, and along with the nanoscaffolds, useful biomaterials, they are prepared into nanomatrices or nanoscaffolds, and they're used for regeneration of bone and also regeneration of skin. Here, hydrogels, adipose-derived stem cells, nanofiber scaffold, nanoparticle impregnated growth factors. How much ever you have your severe burn or accident injuries, that particular tissue and skin can be totally regenerated, making use of the nano-based skin regeneration methods. And also you have hydrogel sealants, like you see in this image, which allow pain-free dressing changes and with patients with severe burns also. So we also have, the major thing is, as we age and if we have even diabetic, we have the dental issues to a higher level. And when you want to replace inflamed teeth or a rotten teeth, now the dental implants are having nano hydroxyapatite, which mimics the natural dental positions. It acts as a natural one. And so the nano HA, that is nano hydroxyapatite, gives longevity and dental implants, which act as a natural dental, it provides and performance and looks almost healing into a natural normal dental implants. Now researchers are also looking for nanoscaffolds for regenerating heart medicine. wherein you can see here, scaffolds are used for guarded cardiac repair. That's a huge challenge now in regenerative medicine. Multi-layered MSCs are taken. You're taken with a sliced biological scaffold. It's prepared, and the scaffold is inserted into multi-layered MSCs, and you're doing the in vitro culture and regenerating the MSC patch. And this is the diseased myocardium. You remove the diseased area there, infarcted myocardium, and implant the MSC patch, which regenerates and then rejuvenates it to the original myocardial patch. So that's how the regenerative medicine is making use of nanostructured medicine. Another new study which is still in clinical phase is how lipid nanoparticles and an mRNA can treat blindness. So researchers have developed nanoparticles which can able to penetrate neural retina into a blind eye and deliver mRNA to the photoreceptor cell and encourage proper vision and develop. And this is in the clinical phase when we can reverse blindness. And that's how stem cells can be used to regrow damaged tissue in many areas of the body, as well as the nanotechnology is taking huge role even in cosmetics. Already in market, many of the nano type of cosmetics are there, especially the nano powder, because of its absorptions and because if it's very tiny nature and having unique properties. One of it which is already in market is your sunscreen, which is very thin layered, and even your anti-aging creams. Nano zinc oxide non-side effect sunscreens are already in market. Instead of having huge patches like that, these easily dissolve and do not cause any side effects. And also, when you are thinking of some transdermal drug delivery, medicine can pass the epidermis, dermis, and reach the muscle. The interior muscle pains can be cured by giving a superficial transderm patch. Like, see, you have motion sickness, your transderm scopulamin is given where you put the patch on your side of the downneath of the ear, and it acts, the medicine is controlled deliver inside into the EM level. And also hydrodermal transmedical patch used for the burns, treatment of burns. And now smart bandages have been developed for controlled release of medicine for all the muscular pains. Dose dependent drug delivery for all kind of muscular image. And this is the one of the kind of new inventions. The electronically it is managed and devised very well, where biomedical application. And here you can see how insight into market progression of various self-healing materials till 2025. These are the various biomaterials involved in self-healing process. So where is the R&D going? From traditional models to nanomedicine, there is a long jump. Now we are looking for the era, we are in the era of personalized medicine. Each person is having his or her type of disease with which a doctor cannot give a generalized treatment. So based on the personal diagnosis, a specialized treatment is given with the intervention of alternative nanomedicine. So nanomedicine is having application in bone tissue engineering, nanostructural implantable materials, as well as diagnostics and therapeutics. So where is India's healthcare? India's healthcare will be revolutionized by nanomedicine. We all know the basmas are the nanomedicine of ancient times. We can, and with that, with the novel kind of Shustruta devised Ayurvedic medicine, we can formulate them into nanomedicine now. One of the important thing is our curcumin from turmeric, a very important medicinal property from Curcuma longa, curcumin, a natural phenol known for treatment you take anything, myelomas to pancreatic colon cancers. It is having a lot of application. So turmeric is the shining star, or you can call as a cornucopia of traditional medicinal plants. It's used for traditional medicine in India as well as China. And what is the major component there? The property is in curcumin. And we know curcumin can cure from your cataract to neurological disease to your urinary tract infection to inflammation of muscle. What is the problem with curcumin is the low solubility and bioavailability. As we take it orally, it is unstable in the gut and it is rapidly degraded. So that's why the bioavailability is not possible. Nanotechnology is encouraged cross-linking curcumin with copolymers of isopropyl acrylamide and N-vinyl pyrolamide, that is VP, or you're preparing pegylated one, formulate curcumin into these formulas. You have curcumin nanocrystals, curcumin nanoparticles, nanoemulsions, all of nanosuspensions, which are acting as with very good bioavailability and curing all these devices. And also another important discovery from nature is Taxol. Taxol is an anti-cancer compound, that is paclitaxel, from the Pacific yew tree, from a Pacific yew tree. And what is the major thing is, again, this we have to take 1 gram of Taxol to cure many of the cancers, you have to cut at least hundreds of the trees. So to make use of this, the Taxol has been used for solid tumors of breast, ovary, brain, and lung. So to make it into a smaller concentration and increase its bioavailability, Taxol is mixed, that is paclitaxel is loaded with PLGA, polylactate glycolic acid, and they are prepared into microspheres and given as controlled release particles, which has been, you can say, the very good natural medicine. One beautiful thing about it is Abraxane is a NAB, nanoparticle albumin bound technology. FDA has approved it exactly 20 years ago, 2005 Jan, for metastatic breast cancer. Nowadays, you're seeing a lot of people are being cured for breast cancer, even in their tertiary stage, because they are being administered with Abraxane, which is a nano drug that is formulated with paclitaxel. So what is the scientist in IIM is telling, Dr. Mishra states, till now we have been giving drugs to patient intravenously, which can get spread to other parts of body, causing side effects. Nanomedicine prevents this. One of the biggest benefits, cardiac patients with blockages in coronary heart disease will get it as nano drugs which easily dissolve the blockage and cure them. So that's where it is taken. And we can say that nanotechnology blended with Eastern traditional medicine and Western medical science integrated can be dawn of future medicine. So that's the goal of nanomedicine, and it has all these advantages. And I had few more slides, but I'll try to push through them. I'll be concluding it. It's been used for various infectious as well as non-infectious diseases. These are the various companies which are preparing various types of nano materials and drugs. And this is the increasing global funding to come up with nano devices for therapeutics as well as diagnostics, cancer, infection control, and cardiovascular disease. This is the various areas of studies and investigational and commercial purpose. And these are the various pharmaceutical industries coming with specifically for cancer therapeutics, Abraxane or Bifin or Nanomedica, Camurus, few of the companies. And from 1950s to 2040, we are come from 1G, 2G to 3G era. 3G era is 2010 to 2020, when targeted delivery in vitro in vivo correlation is done, and we are able to deliver drug based on structure, electric vehicle, and target strategy. And there are various entrepreneurial opportunity also, like smart insulin patch. Instead of giving your various type of syringe-based diabetic medicine or insulins, you have the patches which are not at all painful, like that, even detecting through, like detect your with thermometer, you detect your fever. You can even detect with the nano device the various infections. So that's all the various opportunities with the entrepreneurship programs. And to conclude, nanotechnology offers potential developments in pharmaceuticals, medical imaging, and diagnosis. And though all realization of this beautiful science, it is taking a slow grow, but it's the future of medicine. Nanomedicine is definitely the future of medicine. We need more biofriendly devices to be developed. And hence, we are in the invention and innovation phase and we have to conclude with the commercialization phase. Lot of pharmaceuticals companies and R&D is taking up this role. And at the end, I want to say, let's use this blessing of science for greater benefit of mankind, a suffering-free human race. Thank you.